Yeah, great. Thanks, Jeff, for the introduction, and thanks everybody for being here. Yeah, it's awesome to be here and to see the community that's developed doing tissue clearing and 3D imaging analyses. This community didn't exist back when I was a PhD student, and it would have been really helpful back then to have this. So my lab opened last year at iCORD in the Boston Spinal Cord Center, and we're really interested in understanding spinal cord injury. And so in the spinal cord, of course, there's these long projecting pathways connecting the brain with the rest of the body that mediate motor, sensory, and autonomic function, shown here in green. And after spinal cord injury, these pathways are disrupted, which permanently interrupts communication between the brain and the rest of the body. And unfortunately, after spinal cord injury, these pathways fail to regenerate. And so this disruption of communication that occurs is permanent. And we're interested in understanding why axon regeneration fails. And really, we've kind of broken this problem down into two main factors. First, the neurons themselves. So neurons projecting these axons lose the capacity to regenerate during embryonic or early postnatal development. And we're interested in understanding how it is that developing neurons or immature neurons lose the capacity to regrow axons as they mature. And second, there's this formation of this glial and fibrotic scar at the site of spinal cord injury. So in the schematic, there are astrocytes shown in purple, and they have this really important role of sequestering inflammatory cells and fibrotic cells to the middle of the lesion epicenter. And if we could improve their capacity of these scar-forming astrocytes to do this, it would limit growth inhibition from the fibrotic cells and from the inflammatory cells. And so we're interested in understanding what the signaling mechanisms are that actually enable astrocytes to have this wound healing response. So on the face of it, you would think maybe studying axon regeneration is relatively straightforward, right? You have a pathway that you're interested in, and then you label the pathway, and then you injure it. And then you see at some point later, after some kind of molecular manipulation or a drug or a cell transplantation or a biomaterial, some kind of treatment strategy, whether or not there's more axon regeneration. But although this may seem simple on the face of it, it's actually a lot more difficult than that because it's very tricky to tell the difference between regenerating axons and spared or uninjured axons in tissue sections. You can imagine if you do some kind of spinal cord injury, like in a rodent model, for example, and you've spared some axons, that once you've sectioned that tissue into sections and all you can see are fragments of axons, that it becomes very, very difficult to tell whether or not these axons have actually regenerated or if they are indeed uninjured and never were injured to begin with. And so a few different reviews have been published on this, and I'm showing the schematic on the right. So we have BDA labeled or tracer labeled cortical spinal axons and a spinal cord injury in the middle. And then these arrowheads or arrows are pointing to spared axons that might purportedly look regenerated. And so one of the first applications of the solvent-based clearing method 3-DISCO from Frank Bradko's lab and Alier Turk when he was a PhD student with Frank was basically trying to solve this problem using tissue clearing and 3D imaging analyses. And so 3-DISCO was originally applied to this problem of identifying regenerated axons after spinal cord injury. And what they had done, so they developed this technique with Hans Ulrich Doth, and they had shown that you could use this technique to basically label the entire projections of axons through the entire spinal cord and then reconstruct them in three-dimensional space. And so on the right here, we're looking at some example images of this with a transgenic mouse that expresses GFP and dorsal and sensory axons coming into the spinal cord. And then in the close-up, you can see some of the axons reconstructed back to where the injury site is, shown with this yellow box. And when I was a PhD student, I was really floored by this technique because at the time I was doing a lot of tissue sectioning and reconstructing axons, and I wanted to try it myself. And so then I tried this technique, and I was quite angry because I found that all of the endogenous fluorescence in the tissue that I was using was quenched by this technique. And then I went to Carl Dijkstra's lab to learn clarity for 6 weeks. And I found with clarity that there was this heterogeneous tissue expansion that was occurring that made my tissue into basically an amorphous blob that I couldn't actually do anything with. And so I decided when I was looking for a postdoc, first off, I decided to do my postdoc with Frank Bradka based on his expertise in cellular and molecular mechanisms of axon regeneration, but then that I would try to improve on these techniques for this assay of axon regeneration that I'm particularly interested in, and make it user-friendly and in a way, basically develop a platform that would build on this in a way that other people outside of the lab itself could use. And so in 2019, that culminated in this Nature of Protocol paper where we basically improved upon what Ali or Turk had done in the Bradka lab in a bunch of different ways. And so instead of using a transgenic mouse line with expression of a fluorophore driven by a promoter, we were using adeno-associated viral labeling of axons. Then we were doing whole mount immunostaining of the spinal cord to mark the lesion site. As I mentioned before, astrocytes have this really unique function after spinal cord injury of sequestering inflammatory cells and fibrotic cells to the middle of the lesion epicenter. And so we can do whole mount immunostaining for these astrocytes using GFAP antibodies to mark where the lesion site is. Because when you do this, only the middle of the lesion site itself is going to be GFAP negative. And so you can use this to actually identify where the lesion is. And then we were doing much higher resolution imaging. I mean, if you look at the images here, you can see that only very brightly lit up axons can be identified. And so we were doing much higher resolution imaging, and this is what we're doing now at iCORD. And we developed a few different platforms or methods for analyses using a combination of Amaris and ImageJ. And in the protocol, we do lay out like step by step how to do these things. And so how I spent a lot of my time during my postdoc and now in the first few years of my lab is emailing people who are trying this out themselves and making sure that they're able to do it as well. With the AAV labeling, let's talk about the AAV labeling. So we've also built on this to do not just imaging, high resolution imaging of axons, but of course, AAVs can also label presynaptic puncta, as we've seen with synaptophysin fused to EGFP. And so now we can do labeling not just of the axons, but presynaptic puncta in three-dimensional space. And we have the kind of resolution to do this where we can actually do this across large areas of the spinal cord. And so in my field of axon regeneration, it's not just thought that you could mediate functional recovery after spinal cord injury by invoking long distance regeneration past the lesion site, but that there could be sprouting proximal to the injury and growth of axons and formation of new synapses to form so-called relay circuits. And so we're interested in understanding how different molecular manipulations influence these different modes of axon growth, and therefore developed this technique to look at not just axons based on cytosolic expression of TD tomato, but also synapses based on synaptophysin EGFP. And we have sufficient resolution to do this to look at all of the synapses in three-dimensional space in the spinal cord. And so this image at the bottom marked B is really showing the gray matter of the spinal cord. And you can see these little dots that are white that are basically positive for synaptophysin EGFP. And the softwares that are available, like Amaris, are really quite specialized now or quite strong at quantifying puncta or spheres, as I'll get to later in my talk. And I think that this is a really great resource, especially at UBC, because we're part of this neurophotonics platform. I was just floored by how cheap it is to get adeno-associated viruses from them. I mean, it's like $350 for 100 microliters of a high titer AAV, which means it's much less expensive than even 1 or 2 primary antibodies. And if you couple that with Vector Builder, which is the source, this company that basically somewhat cheaply generates custom plasmids, you can basically get any kind of fluorophore you want generated into or made into AAV particles. And then, of course, with AAVs, there's all these advances in bioengineering new capsids and using different promoters to drive their expression. And so I think as neuroscience has evolved in the last 10 or 15 years, we're getting to a point now where you can basically manipulate any kind of cell type that you want and label them and overexpress optogenetic actuators or chemogenetic actuators. So I'm really excited about that. So for the tissue clearing, I think I'd mentioned before that I had some issues myself during my PhD, but now I feel as though tissue clearing is kind of a solved problem, that there's multiple different techniques that work really well to do tissue clearing. And I tested out a bunch of them at the beginning of my postdoc and eventually settled on cubic clearing with the next generation of cubic reagents. So this is an aqueous-based clearing method where we're using two different chemicals, cubic L and cubic R+, which you can purchase commercially. They're commercially available and they're quite cheap. Cubic L is, of course, a delipidation agent, and cubic R+ is used for the refractive index matching. So it sounded pretty similar to what other people were doing. And of course, the strengths are that it's easy passive clearing. You just put your tissue in there. It's sufficient clearing for smaller tissues, and it's compatible with endogenous fluorescence and immunolabeling. And so if you don't want to do immunolabeling and you just want to go based on what is already like in your tissue itself, I think cubic is really good for that. But it's also compatible with whole mount immunolabeling, which I'll get to the other side on the next slide. But in terms of weaknesses, so we're doing this in the spinal cord of the mouse, which is a somewhat smaller tissue, or we're doing it in the peripheral nerve, which is much, much smaller. And in terms of actually doing this in the whole brain, I don't think that it's a strong enough method. Either that, or you have to wait a really, really long time before your tissue is actually going to be cleared. And it does take quite a bit of time. So we've coupled this with whole mount immunostaining, and unfortunately, this is kind of like the bottleneck, I would say, in terms of getting really nice imaging in three-dimensional space. So we've developed whole mount immunolabeling for unabundant antigens, and essentially what we've done is we've combined this cubic clearing with what Nicolas Renier developed with the iDisco. So iDisco, improved disco, basically took what was happening with the 3-disco method and then added immunolabeling to it. And so we're combining what is basically being used in the iDisco protocol, but with these cubic clearing reagents. So first we do the delipidation with cubic L, and then we go through these steps of primary and secondary antibody administration, and then the cubic R+ refractive index matching. And for some of these antigens, for things that aren't too abundant in the spinal cord or in the brain, like somatics antigens like NUN or CHAT, or if you're looking at specific populations of axons, like serotonergic axons, for example, we've developed pretty good protocols for getting consistent labeling for these across the entire spinal cord. For other things like GFAP, for example, or beta-3 tubulin to look at microtubules and neurons, these antigens are really, really abundant. And because of that, what ends up happening is that you administer your primary antibodies, and then you do this, you go through this whole mount immunostaining protocol, and at the end, you just have this ring of very dense labeling at the surface. And so this is still a problem. And of course, these techniques are very highly dependent on the quality of the primary antibody. So for GFAP, for example, there's a very good primary antibody from DACO. And so we get extremely nice surface level resolution of the astrocytes, but not in three-dimensional space. But for our purposes, it still works pretty well because, as I said, we're just interested in identifying where the spinal cord injury lesion site is. And we can do that quite reliably, even if the GFAP antibody isn't penetrating deeply into the spinal cord. Yeah, I mentioned that the whole mount immunostaining steps happen in between the delipidation and the refractive index matching. So one workaround to this that I was playing around with during my postdoc was to use nanobodies or VHH fragments instead of antibodies. So these are about 1/10 the size of regular antibodies. And this could be one workaround where these nanobodies are much more effective at diffusing into the tissue itself. There might be some problems with the nanobodies that are developed, at least for GFAP. And so we haven't really done very much work on that in that area. And then if you're looking for primary antibodies that are compatible with this whole tissue immunostaining in the mouse, the iDisco website from Nicolas Renier is quite effective for this. Okay, and one thing to keep in mind with the whole mount immunostaining, and really for any of these techniques where you're imaging large tissue volumes, is that autofluorescence is a much bigger problem in the blue-green spectrum than it is red or even far red. And so Nicolas Renier did this really nice study. This is referencing the iDisco paper that came out in 2014, and basically using the same primary antibody, looking at TREC-A in the developing embryo, at E14, and then having secondary antibodies pre-conjugated to Alexa 488 for green, 568 for red, and 647 for far red, you can see that in the green spectrum, or looking at 488, there's much more autofluorescence in green than there is in red or far red. And so you really do want to push the fluorescence antibodies that you're looking at, especially for things that are not very bright, as far red as you possibly can. Now for the 3D imaging itself, we're more using two-photon microscopy than we are light sheet microscopy. And I think for smaller tissues in particular, two-photon microscopy is a really nice alternative to light sheet microscopy. Because it's a point scanning system, the photo bleaching is minimal. And of course, the resolution here, what makes it high resolution is that we're using objective lenses that have high numerical apertures, like 0.8 to 1. And there are some really nice objective lenses out there that have this kind of high resolution capacity or high numerical apertures, but also balance this with a nice working distance where we can image through the entire spinal cord. So 2 to 3 or maybe even 4 millimeters. And then for the imaging itself, I mean, we're using a tunable two-photon laser. So we have an iCORD, we have this two-photon microscope with a tunable laser, and each fluorophore, of course, is stimulated optimally at a different wavelength. And so you have to find basically a wavelength of laser based on how bright the fluorescence is emitting in each channel and what you're looking at, and basically decide from that what it is that you want to do in terms of setting up your image parameters. And then so we stimulate both fluorophores concurrently and then use filter sets to separate the emission. And then here, this again is from the Nature protocol and basically how we mount the sample. And what's nice about two-photon microscopy is that you don't necessarily even need to do tissue clearing depending on what it is that you're looking at, because you can get somewhat deep penetration from the two-photon itself. And so for some of the axons that we're looking at, the dorsal column sensory axons shown here in white, labeled with GFP, it could be that it's not even necessary to do the tissue clearing because they project so close to the surface, the dorsal surface of the spinal cord. So this works not just with the spinal cord, but also in larger tissue sections through the mouse brain. I wanted to show that here. And so this was one practical example of how this kind of technique can be used. There was debate in the literature in the developmental neurobiology field about the role of the small GTPase Rho A in axon specification. And it was thought that if you deleted Rho A in developing neurons, they would form supernumerary axons. So they would basically, their axon specification would break down and they would form multiple axons. And so we deleted Rho A in developing cortical neurons, developing hippocampal neurons, and then sliced the mouse brains into these 1 millimeter sections and then cleared them and then used this two-photon microscopy to look at their entire projections. And basically what you can see is that even with Rho A deleted, there is still only one axon formed. And then we followed this up with some cell culture experiments showing that if you delete Rho A in developing neurons, it has this effect on accelerating axon growth. So their axons themselves are longer, but they're not forming multiple axons. And I think that this is a really nice example of the kind of imaging that you can do using this kind of technique. And so we've seen a lot today about using light sheet microscopy across large areas of the brain. And I think that definitely light sheet microscopy is the suitable technique for that. But if you're interested in smaller areas of tissue and you're not doing tissue expansion or anything like that, then I think the multi-photon or two-photon microscopy is a suitable alternative. So then for analysis, how I learned to do analysis of three-dimensional image data sets is using Amaris. Which I guess is presumably is Arevis's competitor, and maybe because I was in Germany, it was more popular there, but we never really were exposed to Arevis at all, but we're exposed heavily to Amaris. And so Amaris is a great software for doing 3D rendering. You import these image stacks into there. So you have like these tile scan Z-stack images. Of course, a lot of people are using it for reconstructions of the entire mouse brain. And then it's really a pretty user-friendly software for carving your 3D structure into different regions of interest. And so if you want to take your massive image and then basically hone in on some specific area in three-dimensional space is quite straightforward for that. It's quite compatible with ImageJ, and so you can export images from Amaris and then analyze them in ImageJ, which I'll talk about in the next few slides. Generating masks of different regions of interest, so looking at signals in specific regions of interest, doing co-localization analysis. So we're also doing time-lapse microscopy of developing neurons and looking at axon growth and the kinetics of it. And Amaris is really good for this as well. But in particular, it's very effective at doing analyses on 3D structures. And it really, I think, probably its strongest strength in terms of analysis is looking at spheres, spots, or nuclei in three-dimensional space. So I mentioned before that we can do whole matter immunolabeling of for nu-n and quantify basically neurons in the spinal cord. And it's very effective at that. It's also effective at doing calculating volume of signal. So you imagine you have like some volume of axons in three-dimensional space, and then you're looking at specific areas of the spinal cord, or if you're looking at really anything, and you're just interested in the volume of signal that's there, it's quite effective at that. And then finally, it's very good at making fancy videos. So how we basically developed a couple of different pipelines to do axon growth analyses in three-dimensional space using Amaris and ImageJ. First, a manual analysis, and then a semi-automated image analysis. For the manual analysis, and again, this is all step-by-step spelt out in the protocol. Basically, what we're doing is we're partitioning the three-dimensional structure into different regions of interest, 200 micron blocks relative to where the center of the lesion site is. And then we're manually annotating axons with little dots, and then using that to normalize for the levels of sensory axons that are labeled to begin with. And we use this when the density of the labeling is not too high. We can also use a very similar technique, as I said before, to measure the total volume of axons within the area. For semi-automated analysis, basically what we're doing is something somewhat similar, but then we're exporting this three-dimensional structure, this 3D rendered block of axons in Amaris into a series of rostral caudal cross sections. So now the line that is the axon in the longitudinal plane is a dot in the coronal plane, and then you can quantify in ImageJ the number of dots. And this provides you basically, as I said, it's semi-automated, but of course the trajectories of regenerating axons are quite tortuous and random because the guidance molecules that exist during embryonic development to guide them to the right place to form synapses are gone. And so if an axon bifurcates or if it turns backwards, then the semi-automated analysis is not effective at dealing with that. So in terms of applications, as I said before, we're interested in understanding why neurons fail to regenerate after spinal cord injury, and we're focusing on primary sensory neurons and basically trying to identify genes and intracellular processes that prevent axon regeneration. And just focusing on these primary sensory neurons, we're using transcriptomics to compare gene expression changes in primary sensory neurons, or DRG neurons, in 3 different conditions in which their ability to grow axons changes to try to identify what genes are suppressing axon growth. So in cell culture, these adult primary sensory neurons start off with this very compact level of axon growth, multi-branched, arborized mode of axon growth, but by 24 or 36 hours in culture, they more than triple their axon growth velocity and have this elongated growth mode. During embryonic development, there's these 2 different phases where they're rapidly extending axons but haven't reached targets yet, and then when they're arborizing and forming synapses in the spinal cord. And although these neurons fail to regenerate after spinal cord injury, they are pseudo-unipolar morphology with an axon that bifurcates close to the cell body, projecting one into the spinal cord and the other peripherally. And after peripheral nerve lesion, they do have this ability to regenerate. And so we reasoned that if we did RNA-seq across these 3 different paradigms, looking at gene expression changes that were common across all 3 paradigms, that we would be able to discover novel genes and intracellular processes preventing regeneration. And long story short, in this paper that came out a couple of years ago, I identified that there's a common set of synapse-related genes that are negative with transcription that's negatively associated with axon growth competence. So with prolonged cell culture as well as with peripheral nerve lesion, these synapse-related genes are downregulated, but with embryonic development during this phase, this transition from axon growth to synapse formation, these are massively upregulated. And there is an enrichment for genes encoding proteins related to ion-gated channel activity and to snare binding, in particular genes related to the presynaptic active zone. This led us to eventually collaborate with Dr. Niels Brose's lab to investigate the role of MUNC13 in axon regeneration, as well as other active zone proteins. And we particularly focused on MUNC13 because it has this very special role of being essential for synaptic vesicle priming. And so in the absence of MUNC13, or if the neuron is devoid of MUNC13, the synapses are no longer readily releasable, and there can be presynaptic calcium influx, but there's no actual synaptic vesicle release. And so you could use this as a model to block neurotransmission and see what kind of effect this has on axon regeneration. And if you block transmission, this enabled axon regeneration. And so this is shown in the bottom panel. We have these GFP labeled sensory axons and the spinal cord injury marked by the asterisks. And you can see that axons are regenerating right through the lesion site itself. And in the lab, what we're studying now are basically the molecular mechanisms underlying how MUNC13 blocks axon growth, with the particular focus on cytoskeletal dynamics at the growth cone. The other application of this relating to the astrocytes. So when I was in the Bradco lab, there was this project going on that basically was trying to understand how extracellular inhibitors that are present in the adult environment are actually suppressing growth in neurons. So there are multiple molecules present in the environment of these neurons that are suppressing growth, such as chondroitin sulfate proteoglycans and central myelin, but it wasn't entirely clear how it is that these molecules actually shape axon growth, or how this instructs axon growth suppression. And we found that how they do so is by basically activating RhoA within neurons, which ends up activating actin compaction at the growth cone, which suppresses microtubule protrusion, suppressing axon growth. And so if we deleted RhoA, this didn't have an effect on, and then cultured cerebellar granule neurons on both growth inhibitory and growth permissive substrates. On a growth permissive substrate, there's no difference in axon growth, but if you plate these neurons on CSPGs, this restores the ability of these neurons to grow axons. But if we tried this in vivo after spinal cord injury, this does not promote axon regeneration. But the way that RhoA was deleted in this study was using a nesting-Cree promoter driver line, which basically doesn't just delete RhoA in neurons, but also in astrocytes. And you can see in the image here, so GFAP is shown in red, and you can see that there's this enhancement of GFAP immunoreactivity in the vicinity of the spinal cord injury lesion site. And so because the RhoA was being deleted in astrocytes, and because we saw this phenomenon of this increased GFAP immunoreactivity, this suggested that perhaps RhoA controls astrocyte reactivity. So then I engaged in a study where I basically deleted RhoA in neurons versus astrocytes versus both to see what effect this would have on axon regeneration. And here, this is how we did this. So looking at the nesting-Cree RhoA knockout, there's no difference compared to control. But with GFAP, so RhoA deletion specifically in astrocytes, there was more axon retraction or degeneration. Whereas if we deleted RhoA specifically in neurons, we had potent axon regeneration. So this is a video on that. And so we have the spinal cord injury lesion site, and this shows the kind of images that we can get from MRIs. And you can see that there's very limited regeneration in the control situation. Then in the nesting-Cree RhoA knockout mice with RhoA deleted both in neurons and astrocytes, there's also very limited regeneration. Then in the astrocyte-specific RhoA knockout, we have more axon retraction and expansion of this non-neural lesion core that's GFAP negative. And so there's retraction of axons from the lesion site. But with neuron-specific RhoA deletion, we have this nice regenerative response where axons are able to make it past the lesion site due to their lack of responsiveness to these extracellular growth inhibitors. Okay, so now we are focusing on a few different things. I mean, we're still interested in understanding, we have follow-ups to those projects going on currently. We're also developing a tissue clearing pipeline to assay peripheral nerve regeneration. And so, as I mentioned before, we have these neurons don't regenerate after spinal cord injury, but they do after peripheral nerve injury. And so we're developing a complementary assay to look at peripheral nerve regeneration in three-dimensional space based on immunolabeling for SCG10, this marker that gets turned on specifically in regenerated axons. And we're also interested in doing retrograde labeling of the supraspinal projectome. So basically all of the neurons that project to the spinal cord, and then using retrograde labeling to basically identify neurons that are the most responsive to spinal cord injury to see which ones are actually mediating recovery of function. Okay, that's it. Yeah, I want to thank you all for being here and everybody that's in my lab and the funding sources, and I'm happy to take any questions.